I'm a faculty at the Department of Physiology, Biophysics. So I've been here for 15 years, and my research focuses on developing mass spectrometry-based proteomics approach to study different types of systems and protein complexes.
Can everyone hear? Is the mic?
Can you hear me at the back? Yeah, okay. So during the last 15 years, my lab actually has provided different kinds of services to people on campus and off campus. And so recently, with the help from NIH and also the support from the school, we were able to obtain a high-end instrument called Orbitrap Fusion Lumos Mass Spectrometer. So this instrument is the state-of-the-art instrument with the innovative technology combining 3 different mass analyzers and provide the flexibility, sensitivity, and for the proteomics analysis. So basically, if you compare to different kind of instrument, this instrument provides the best performance for low-level analysis down to entomole-level peptide analysis, and then also has the flexibility to perform different types of quantitation, advanced post-translational modification analysis, and also structural characterization and for intact protein analysis. So with this capability, it actually also provides the capacity for us to be able to serve more people. So then we use this instrument and established a core facility recently, means that we will start the charging system and then to provide the advanced proteomics services to the entire campus. So based on the capability of the instrument, we will be able to provide the services as listed listed here. So we will be able to perform the analysis and to do the fast and deep proteome profiling of the whole proteomes. And we will also be able to provide different capabilities for different types of quantitative proteomics. And we also will be able to, as I mentioned, to study various kinds of post-translational modifications. And due to the capability of the instrument, have different types of fragmentation techniques. And then we will also be able to do the structural analysis, which is related to the technology we developed in our own lab. So this will be a special service provided only by our lab. And also to do the intact protein analysis. So for the remaining time, I will start to give you some examples of the analysis related to these different categories. And also later on, I will introduce Clinton, who now is the facility manager, and to perform all these analysis and then provide the services to you. So for the proteome profiling, with this instrument, you actually can really do very fast analysis to generate a profile of around a couple thousand, and could be more than 5,000 proteins in a single shot. And so what you do is that you actually have different types of samples can be carried. The samples can be from the cells, tissues, and the body fluids. And those samples can be digested. The proteins can be digested and subjected to the mass spectrometry for analysis. And then after data analysis, then you can generate those proteome profiling, which can be used to correlate with other types of profiling obtained from genomics or transcriptomics. And so this way, you can combine all different kinds of omics information and then to try really understand the cellular process. And specifically, I think we're all interested in discover biomarkers and for different types of disease for early diagnosis or improve the treatment. So here I listed the example for possible projects, and you can look at the whole proteome, and also you can look at the partial proteome, the subproteome by subfractionation, and to get organelle proteomics. And then you can also do conditional dependent proteome profiling, including to do signaling and to do drug treatment or different developmental stage, and to look at the proteome profiling. So this gives us the capability to look at the larger scale of the proteomes, which we were not able to do in the past. And so in addition to protein identification, the quantitative information is important in biology. And for that, we can provide different types of quantitative analysis. These include that the label-free protein quantitation and isotope labeling-based protein quantitation. And so isotope labeling-based protein quantitation involves either using metabolic labeling or use chemical labeling. So all these different kinds of analysis can be performed using our instrument. So for label-free quantitation, it's kind of similar to other types of analytical tools that you can do label-free analysis. So in here, basically, you have multiple samples. You can digest the proteins and subject to mass spec analysis, and then use the mass spec information to do the quantitation. So these allow you to compare different samples from almost, you can say, unlimited number of samples. You can do as many of analysis as you would like to. And also, it's good for monitoring dramatic changes among different samples. And definitely, there's no additional cost in your sample preparation, so relatively easy. However, it does have some disadvantage, because if you have 100 samples you want to compare, that means you actually have to analyze the 100 samples individually by instrument. So that means you will be charged for 100 times. So, and and also because you analyze sample separately, sometimes it requires such a high reproducibility among the experimental conditions, like the liquid chromatography has to be very reproducible, mass spec has to be very reproducible, and so on and so forth. But nonetheless, this is a very easy way for you to start to do some kind of testing and optimization of your procedure. So for clinical analysis, we like to analyze large cohorts, and we also like to analyze 100 samples. When you submit a grant application, people always say you do clinical biomarker discovery. How many samples are you going to analyze? What is the statistical significance? So for that, it's important to do multiplex quantitation. And so currently, that there are reagents available through Thermo Fisher Scientific to allow you to do up to 11-plex. And that means that you can compare 11 samples in a single experiment. And so these will improve the throughput, and also you improve the quantitation accuracy, and as well as have less missing values. What does that mean? It means that if you have different proteomes, say if you have breast cancer, you have different types of cancer cells, then you can do cancer type 1, the cell type 1, 2, and 3. Then you combine them together. Maybe some protein present in one type of cancer types, the other present in another cancer subtypes. So this way you can see the whole proteomes present, and then try to figure out the networks of this information, then to allow you to better characterize and the proteome and figure out the biomarkers. And so definitely the reproducibility always be improved. Even if you don't have hundreds of samples, you could also to do your experiment to improve the reproducibility. Say if you have 3 experiments, I could do the first set 3, second set 3, and third set 3. Then I combine them together, so I have all my biological replicates required by NIH in a single experiment. So in addition to relative quantitation, you could also do targeted quantitation. So this is a definitely new feature I'm very excited about, because we didn't have the capability before. And what does it do? So as other people sitting in the room, probably you have all done Western before. So this is you can consider as a mass spec Western. So what does it mean? Instead of using antibody, you're using peptide signature. And those peptide signatures from each proteins you are interested in. So you can choose 300 targets and say, I'm very interested in these 300 proteins, and then I monitor them specifically. So this targeted analysis allow you to improve the sensitivity 100 times at least compared to routine analysis. So this way you can really dig deeper, and then to be able to screen those targets. So then you can generate information like a microarray kind of. So this indicates that the intensity of different peptides correlating to different proteins. What this information can do for you. This information can allow you to do biomarker discovery, validation. So you first discover the biomarkers, potential biomarkers. Then you have set of targets. Then you try to quantify them, and then because you can explore them at a very low level. And then you can also examine the protein abundance differences and their changes through different conditions. And so, for example, during the aging process and during the tumorigenesis and so on and so forth, and then to allow you to see which group of proteins actually change. And then you also could allow you to do quantitation on the modification patterns and correlate it with the protein abundance. And of course, it can use it for protein-protein interactions. And now currently, I think there's a popular saying about network biomarkers, not just one. And with this, you can also calculate the protein absolute abundance. And this information could allow you to determine subunit stoichiometry of the protein complex you're interested in. So this new capability actually provides so many opportunities for you to explore the different types of experiment. So we all know that the proteins not just have one single form. They have multiple forms due to different types of post-translational modification. And these post-translational modifications are actually very important in regulating their function and the structure. And there are more than 300 post-translational modifications have been characterized so far. And so those are the common ones I listed here, including phosphorylation, ubiquitination, acetylation, and glycosylation, and many others. So when you analyze the proteome, you can look at the entire protein expression level. You can also look at the specific post-translation modification subproteome. And currently, there are many different ways you can enrich those subproteome through antibody enrichment or through affinity enrichment and so on and so forth. So as listed here, if we try to look at the phosphoproteome and the ubiquitin proteome, and then we can analyze the peptide and enrich for these specific peptides and to do the analysis. You can look at those at the global level. You also can look at the specific protein level, or you can also target it to quantify this and then to understand their dynamics throughout the different cellular process. So the next would be about the protein-protein action structure characterization. I will let Clinton finish the rest of the talk because he really has contributed significantly to this part of technology development. Yeah. Well, we try to cover as much as we can because we are so excited about this new facility and how you can provide service to you.
Okay, thanks for the introduction. So as Lan suggested, this is actually one of the things I'm most excited about offering because this has actually been one of the primary focuses of our lab for the last few years. And this, of course, is cross-linking mass spectrometry for structural analysis and for PPIs, protein-protein interactions. So I'll kind of give a brief introduction to what it can actually offer. So typically, people can use mass spectrometry to study protein interactions by coupling it with affinity purifications or immunoprecipitations. So one of the main caveats of this approach is that it can be difficult to biochemically validate whether the interactions between the bait and the prey are actually binary interactors or not. So we've developed, I don't have a lot of time, so I guess I will just say that we've developed these chemical reagents that essentially covalently link specific residues together if they're within proximity to each other. And so what that actually ends up creating is that we can develop these more interactome maps that suggest proximity between proteins. And that also gives us clues about which proteins are actually coming in contact with each other and how they actually function. So we've applied this strategy to a multitude of different protein complexes. In our lab, our main focus is using cross-linking to understand proteasome structure and function. And so using cross-linking, we've been able to probe the topological interactions between the subunits within the proteasome, as well as some novel and known interactors as well. So we and other labs have also applied this kind of cross-linking workflow to study protein-protein interactions at the systems level. And so this can be used to get an idea of what are the channels of communication between different protein systems. So as Lan mentioned, a lot of these proteomic approaches can be coupled with quantitation to provide another layer of information. So we can actually quantify the changes in protein-protein interactions, and these condition-dependent dynamics can have influences on the interaction network, but they usually have some basis on the conformation or the architecture of protein complexes as well. So these conditions can result from post-translational modification interactions or protein-protein interactions, other types of environmental stresses. So previously, we published a paper using stable isotope cross-linkers to probe for the differences in SCF ubiquitin ligases that accounts for their inactivation by a bacterial effector protein. Recently, we also published a paper detailing a workflow for multiplexed quantitative cross-linking as well. In general, the workflows and the platforms that we've developed for cross-linking are actually really applicable to a wide range of interactions, and they're including but not limited to antibody, antigen, host pathogen, and ligand receptor interactions. As I mentioned before, they can be used to study the topologies and structures of macromolecular protein complexes, as well as for proteome-wide profiling of protein-protein interactions. They can be coupled with quantitative analysis for comparative analysis between different conformations of proteins. And I will also say that one of the main pros of such a versatile approach is that compared to other structural biologist tools, we're actually able to get a glimpse of how proteins function and their structures at a more biological level than most. So this cross-linking can actually be performed on cells and tissues as well. So the last thing I'll talk about in terms of workflows is another service that we plan or we will be offering, which is the idea of intact protein or top-down analysis. So all of the workflows that Lan and I have talked about so far constitute bottom-up proteomics, and that's to say proteins are first isolated and they're digested into peptides, and then these peptides are analyzed by the mass spectrometer. And then using database searching, we can identify the peptides, correlate them back to the proteins, and identify what the protein content in our samples are. So this is really important for several reasons, but in the context of post-translational modifications, it's important because it allows us to map the exact sites that residues are modified, and also allows us to quantify how often those sites are modified. But there is some detail that is actually removed when these proteins are digested into peptides, and as Lan said, there are hundreds of different post-translational modifications, and each one can actually happen simultaneously on a protein. So that information is lost when all of the proteins are digested into peptides. So as an alternative, top-down proteomics extracts proteins usually by way of purification, and these proteins are actually submitted to the mass spec directly for analysis. And what this analysis allows us to do is it actually allows us to measure the intact protein masses for all the different forms of the protein. It actually allows us to capture all of the different proteoforms of all the proteins with all the different combinations of post-translational modifications as well. So that's also something that we're really excited in. So most of the slides that we talked about detail different types of mass spec analysis, whether they're tandem mass spec or multi-stage mass spec analysis. But we will also be offering a variety of options for sample preparation. This includes primarily protein digest, and depending on your complexity of your proteins, could range from in-gel protein digest for simple proteins to various denaturing in-solution digests for more complex samples, such as cell lysates or cell pellets. We also offer a variety of data analysis options for protein identification and quantitation as well. And of course, we also offer customized packages for cross-linking as well as more complex types of quantitation. Feel free to contact us for consultation regarding experimental designs and planning out your projects. We also offer training for sample preparation, such as protein digests and protein purifications, extractions, and routine data analysis and interpretation as well. The Department of Physiology and Biophysics also offers a course for proteomics every winter quarter for anyone that's interested. And in the future, we also plan on hosting some future user meetings to help disseminate new proteomics technologies and to make mass spec analysis more accessible for everyone. And so with that, this is a temporary link to our website. It's also linked to the Biophysics department website as well as Lan's faculty site. And so I guess I'll take any questions if you have them. Thanks for your attention. Yeah.
So where are the facilities located, by the way?
So the facility is actually located in our lab, and that's in D224 Medical Science 1.
Okay, so I know the chemistry department also has the mass spec facility. So I mean, do you guys coordinate, or who and when should do yours versus theirs? So it's always good to try to get a sense of how.
I think
I might ask Marion to address that actually.
I think Rick meant this year, but why don't you answer Rick's?
so like, should I say it or should Rick say?
Well, I hope it's not a hard question. I mean,
well, no, we envision and with the Office of Research and Suzanne, the School of Medicine, Syed, standing up actually a multi-site mass spec shared resource. It has different machines at different locations and maybe a focus on different primary capabilities at each site. So we actually convened an external advisory board last year, 3 outside scientists to look at this for us. And the major recommendation that came out of that was we needed to get a high-end machine. The Lumos Tribrid is it. So everybody put skin in that game, and we were able to acquire it. That component is going to be based with land. And you heard about that today. We also have the chemistry facility, which has a water Zevos machine that may be more suited to
Metabolomics.
things like metabolomics.
And they're also focused.
And then Publishing and Syed also has a Thermo Fisher machine that's more suited toward PTMs, I think. So we intend to try to stand up a matrix shared resource that would be sponsored in part by the Cancer Center. But a big part of that would be consultative services, so the user can decide which branch of that would be best suited for their particular experiment. So Marion and I are working, and Suzanne are working hard on that now.
Obviously a timely question.
So just a really practical question. So who runs the samples? Do we just give you the samples and you run them, or do we have to run them, or how does that work?
So at the moment, Clinton is it. And so I think in a way, the best operation right now is that we train you how to do sample prep. And then you provide the final digest, and then we can run the sample, analyze it. And this will also minimize your cost. So there are people asked us whether we could do from very beginning to the end. We can also do that. Basically, we just charge you more.
but can someone do everything themselves if they're trained? Is I guess the opposite question.
I think it's practical for people can do the preparation, like to the level of the peptides. Yeah.
But that's really the crux thing here, Lang, is to what extent is there a steep learning curve for the user that's preparing the sample? Because the last thing that you want to end up with is somebody reading a cookbook, preparing the sample, and then having all your time and their time and money wasted because the level of sample is not good enough for
yeah, I mean, there's a diversity in the sample preparation. I totally agree. I think for the lab, it's also depending on the lab expertise and how well they can prepare samples. So what I can say at the moment is that we will provide the most robust protocol, and then we will train you diligently, and then we hope. Because in a way that I think through time, if we get more help, definitely. And also I talked with Suzanne, we are in the process try to get a seed instrument to do robotic digestion, this kind of stuff. So I think if we are successful, we could process 100 samples at a time. This way will minimize the variations in your sample prep, maybe more robust. So all these requires the time and also the support from you, as well as the reality how we can get this kind of service.
Could you comment briefly on the other end of the analysis? So what kinds of software could be available to users, or is that all to be handled in-house?
So we have actually couple different kinds of softwares. So there's two softwares actually we use actually are publicly freely available. And those ones, the users, they are welcome to download it in their own lab and then to try to have more kind of analysis. And in general, we will do the data analysis and then provide the format to you, which you can visualize, and also to allow you to submit for publication or for grant application.
Okay. Your transgenic mouse facility or genome engineering facility on campus. If you want to do anything or need any advice with regards to using genetically engineered mouse models or making them in your research plan, then the TMF, Transgenic Mouse Facility, which is now also known as the Mouse Genome Engineering Facility, is the place to go. There are many commercial options, but I would encourage you to try us first because we provide a real white glove service, and you'll find our pricing is very competitive, if not better than commercial services. If you simply Google UCI TMF, it'll take you to our homepage. There's lots of information on the home page, but the key thing that you want to probably pay attention to is this here. If you have a question, just click on that link and email us, and we'll get back to you in a timely manner with a response that will hopefully provide you with what you need. The TMF MGEF is organized under ULAR, which most of you will know about, University Laboratory Animal Resources, and that in turn is organized under the Office of Research. So we're kind of school agnostic, although we're physically located in BioSci, and as you'll see, we enjoy support from many different entities on campus. Okay, so who are we? Sherry Collins is our colony management personnel. She looks after all your mice as they're being generated and biopsied, etc. Shimako Kawauchi is a relatively recent recruit. Shimako is a project manager for a large Alzheimer's program project at U54, and she also helps to provide support for the TMF on a variety of other projects. John Newman is our managing director. He took over from Tom Fielder about 4 years ago now. John is your initial point of contact. If you click on that email, the TMF, he's the person that's going to get your email, and he will respond to you or contact me or Shimako or someone else for information. Kaixuan Shi, he's rather shy. He tends to play with all the mice. He's actually the person that's going to be doing your microinjection or electroporation of embryos, as we're now starting to move towards. And then Shuling Wang is a multitasker. She continues to do ESL-based projects. She'll do all the Southern analysis. It's remarkable how few people can do as good Southerns as she does. Right now, we're doing some for a cancer center investigator at UCSF, for example. So we get lots of samples from around the country still to provide this type of service. And Shuling also helps out with many other aspects in the TMF. So what do we do? As the first bullet states here, if you have any question about genetically engineered mouse models, just ask us. We implore you, if you're thinking about getting into this, to contact us early. Okay? And we can give you informed advice about the pitfalls, and there are many, of making and analyzing these types of mice. In many cases, for example, we can actually find the mouse that you want without actually having to make it. Okay, it's quite challenging to find existing mouse models on the web, but we have ways of tracking these things down. As most of you, if you haven't been living on Mars, will know, CRISPR-Cas9 is all the rage now. In the past 2 or 3 years, the TMF has changed over to using CRISPR-Cas9. Recently, we just passed our 50th project that's completed, and these are a wide variety of things, from point mutations to knocking in coding sequences. So you're pretty much limited only by your creativity as to what you might want to do with the mouse. Nonetheless, we also provide transgene and back integration services, including single copy backs. We are a core facility for another program project at Cornell University, generating backs to look at optogenetics in the brain. So we've got a lot of experience in analyzing backs. We have a pulse field apparatus for a CHEF mapper for doing these sort of things as well. We can source ESL lines that have gene trap mutations or other sorts of mutations and generate mice for you from those ESLs. And again, we can source, import, cryopreserve, and store for you, do IVF to expand your colonies, and export your mouse embryos and germplasm to other investigators. So for the Cahalan lab, for example, they have a very successful mouse model that's used to look at calcium imaging in mice and in real time, and that mouse has been developed in collaboration with them, and Mike just told me it's recently been exported to Jackson Lab, who are now offering it for sale. So that's the sort of soup to nuts type service that we offer. Letters of grant applications, yes, and I'll get you yours later today, and much, much more. Okay, so this might surprise you. This is who we support. This is a map of institutions, not just investigators, but institutions throughout the US. We've even had inquiries from New Zealand and Australia and places overseas. But there's over 50 institutions that we've provided support for since the facility was developed. And many of these, as you can see by the orange dots, are multiple use customers. So people that come back to us again and again. 2 or 3 years ago now, we actually became the default transgenic mouse facility, genome engineering facility for UCLA. So many of you may not know this, but we actually do more work for investigators at UCLA than we actually do for UCI. We also service Riverside, and we pick up a lot of people from UCSD as well. So the bottom line is that we have a lot of users. Routinely, we have maybe 60 or 70 projects a year, and so it keeps us all quite busy. This is all made possible in part by recharge from the users, but also by extremely generous support from the Office of Research, BioSci, School of Medicine, and from your cancer center. I'll just put a quick plug in for a relatively new bit of equipment that we got, because this might be of use to you. In people that are trying to engineer Cas-type mutations in cell lines, one of the first things that you task your poor graduate student or postdoc or undergrad with is, oh, why don't you just go ahead and screen those 400 clones and see which ones might have a change in them. Okay, and the person rolls their eyes and says, oh God, I'm running gels forever now. Okay, so because of this sort of aspect in the TMF, we obtained a fragment analyzer, similar in ways to ones that's in the GHTF. But what this allows you to do then is to load up 3 96-well trays and wander off home for the night. And this will quietly perform gel or capillary electrophoresis and give you very nice graphic and quantitative analysis of fragment sizes. The resolution we've set up right now is 5 bases, but it can go down to 2 bases. So if you want to do a rapid screen to find out, do I have any of my clones that I've transfected with my Cas9 ribonucleotide proteins, etc., are there any that could be a candidate? You can use this to find that out. And if anyone's interested in using this, just contact the TMF and we'll get you set up to run overnight samples. Okay, so I'll stop there. My 5 minutes I see is up. And
Adeela Syed from the Optical Biology Core is our next speaker. And I wanted just to give another shout out to the Office of Research, the schools, and the Cancer Center for their support of these shared facilities. So the Cancer Center is coming up for its renewal in 2020. This year, right now, starting in a month or so, is the year that starts to count for all the metrics that will be submitted. So please remember when you're publishing papers with these wonderful technologies to cite the Cancer Center support grants. Adeela Syed is the capable manager of the Optical Biology Facility, and she'll be telling us about a technology that has caused a super lot of controversy. Maybe she'll go into that a little bit, but also excitement.
So thank you. I've been around UCI for about 20 years as a postdoc and a research associate, but I've been managing in the OBC for the past 6 years. And during that time, I've seen the delivery of 3 confocals and 1 light sheet, and just like we have with kids, I don't have favorites, but the light sheet is our newest microscope, and so I'm most excited about that for now. But Suzanne, I'd love to come and tell everybody about all the stuff the confocals can do as well, to be fair. Okay, so light sheet microscopy is called single plane illumination microscopy. It's got a couple different names, but we'll just call it light sheet for now. So the first question is, why do we even need a light sheet microscope? And so typically people will have in their labs a wide field microscope, and the wide field microscope is great because you can illuminate everything in the XYZ field, have a camera, and take an instant picture. The problem comes with light sheet is that if you get anything thicker than 5 micron resolution, you start to get all the out of focus light coming into the picture. So that's where our confocals come into play. And the confocal uses a physical pinhole to block out the out of focus light, except now instead of a camera, we're going to use a detector and scan our way through the entire image, which obviously is going to take longer. We're also eventually going to be limited by how thick the sample can be before we just start getting too much scattering. The other downside to confocal is that you're mounting your sample between a slide and a cover slip. And so eventually you're going to have to squish a sample that's, like a 3D sample, like a brain or a heart. And if you do want to look at a whole organ, you're going to have to slice it all, image all the slices, and try and reconstruct it all together, which is what something an undergrad, I guess, would do, our graduate student, but very time consuming. Okay, so the need for a light sheet was to be able to look at large tissues And the other thing too is looking at live animals like zebrafish imaging or something, you run into the problem where even though you're only looking, okay. So when you're looking at a zebrafish or something, even though you're only looking at a small section in XYZ, the laser is actually going through the entire sample. And so you run into the problem of phototoxicity for live imaging of animals, especially over long periods of time. So we ended up, after a very intense discussion, to purchase the Zeiss system. And so I do want to spend a couple minutes explaining why we purchased this system. It ended up being like a Goldilocks situation where it was just the best one in between. There's 3 commercial systems available out here. There's a LaVision, a Zeiss, and a Lucendo. And let me just go briefly over 2 of them. We were able to demo the Zeiss and the Lucendo in the OBC. You can see if you just look at resolution, the Lucendo has probably the best resolution, but on the other hand, it has the smallest working distance. And so if you want to look at a whole brain, that's not going to be the optimal system because the brain wouldn't even fit. And so this is the Lucendo setup down here. The sample would have fit right there. The LaVision was in the opposite end where it could accommodate the largest sample. The problem is if you looked at the acquisition for the frames, you could only acquire 4 frames per second, so people doing live imaging, that wasn't practical. The Zeiss system was literally quite in between. We were in between in terms of working distance, and we were actually pretty good in terms of the acquisition speed. The downside and the holdback to buying a Zeiss system was, as good as it was, there was a lot of clearing of tissues using this organic clearing method, and we were having 2 problems. One, we weren't able to necessarily get good imaging with that, and so Zeiss has actually worked in a collaboration now with UCI to help develop and fix this chamber and this light sheet offset. And the other thing came is that if we wanted to look at a whole brain with the Lucendo system, you could have looked at the whole brain without doing any tiling or stitching, which wasn't going to be possible with the Zeiss system. Zeiss promised and eventually delivered a 2.5x objective, and now we can look at the whole brain without tiling. So if you, just an example, want to look at a whole brain, it takes about half an hour, right, for 2 different fluorophores. That being said, most people do tile. Okay. So really the initial data, and I teach a microscopy course, and I would always joke and say if somebody gave me $500,000, I would buy a light sheet microscope. And the last time I taught the class, I said, somebody gave me $500,000, we bought a light sheet microscope. But really, the money and the initial push came from work done in Sunil Gandhi's lab, where they literally took their brains down to Caltech, imaged them on the LaVision system, got some incredibly beautiful data. And I say Dr. Gandhi's lab, but it's really his student Ricardo, who's been just amazing and a great source to have at UCI right now. And like I said, we have this collaboration now with Zeiss as well. But funding for the microscope was really thanks to the Vice Chancellor of Research, big thanks to Brandon for this, and School of Biosci, School of Medicine, and the departments of Cell Biology, Neurobiology and Behavior, PharmSci, and the Cancer Center, and Learning and Memory. Okay, so light sheet. What is the difference? So the difference now is in a wide field microscope and a confocal microscope, you're going to shine the light through your sample in the same direction as the objective. So that's why everything in your light path is getting illuminated. With the light sheet, what you're going to do is all the lights are off and you only have light going in one Z-plane, and so when you can now use a camera, take a picture so it's faster, and you're only seeing where the light is being shown. Okay, so you have these. This is the Zeiss setup right here, where we have an objective on either end over here. The objective is shining a light perpendicular to the objective, which is going to be acquiring the image over here. Just like with anything, if you shine a light on it, you're going to get a shadow. And so to avoid shadowing, the Zeiss system is set up, has 2 different ways to do this. One, the lights will literally pivot really fast, and so that helps get rid of the shadowing. And the other thing is that you can just excite the light from both ends. And so whatever shadow you get on one end, you kind of bypass that on the other side. And so watch really quickly, because this is going to be a video or animation that plays. There's a little zebrafish sitting in here, and it's going to go through the light and light up. Okay. Do you see it? I didn't set the timing on that. It's from Zeiss, but I can do it again. There it is. Okay. So there's an automated stage that'll literally physically move the sample in Z so you can generate your Z stack. And it's very, very fast. And like you said, we can go up to 67 frames per second for 2 different channels.
Are you going to show us the image that was captured?
I am. So Zai sent me all these beautiful images, and I said, you know, I'm going to just show images acquired at UCI. So some of them look great. Some of them are not as great, but I didn't generate the movies. They were generated by the person who collected them. But shout out to all those people who were I was on their case. I need a video. So good for them. So not every sample, obviously, is as easy to work with as a zebrafish embryo, which is a clear animal. Most tissues are opaque, and you have all this refractive mismatch, which causes a lot of scattering of light. And so if you do that and try and throw a brain or a lung or a heart, all things we've looked at now in the light sheet microscope that we have, there's just a lot of scattering of light. And so you need to go ahead and start clearing tissues. And this is probably going to be the most time-consuming part of doing a light sheet microscope, is going to be sample prep. And so there's different optical clearing methods that are used to clear samples. And so here's an example of using scale right here. So this is a mouse embryo, and so some people are using scale right now for doing embryos and for doing mammary tumors. We also have clarity, which is an aqueous clearing method, and this is iDisco. And in this tube, there really is a brain. And so different clearing methods are going to be good for different situations. If you have endogenous expression that you want to make sure you keep, you don't want to use an organic clearing method because you're going to quench your fluorescence. But if you're going to use an antibody, then this iDisco method, for instance, works really, really well. Okay. Any questions on clearing? Okay, good. So types of samples. So Suzanne, this is show and tell, right? So, okay. I always find it easiest to try and explain something. This is what the actual chamber looks like that you're going to put your sample into, right? And
Do you need more lights?
no, I'm good. Everybody can see it, right? So there's windows on all 4 sides. The illumination objectives are going to be mounted on both sides right over here. You're going to have your detection objective on the back end over here. You're going to put this into the chamber, and then your sample is going to be suspended down in the center over here. And there's no eyepiece in which to focus your sample, and so the way you focus your sample, you close the whole system up, and there's a camera over there, and then you use the back end light to get your sample in position. So we can hang the samples. So this is a brain that's hung, and this is a front view, and this is a top view, and you can see the light coming through and just illuminating a small section over here. You can embed it in low melt agarose, and so we have syringes and capillaries that we will provide to you. And so you can have, if you have small little embryos or small ovaries or whatever it is, you're going to put it into the low melt agarose, suck it up into the capillary, and then add the capillary into your sample and plunge the agarose into the chamber. And then for people who need really, really low melt, really, really low percentage of agarose, we actually have FEP tubing, and so you can create like an agarose plug on either side and suspend your sample in the center. So here's an example. The brain is suspended over here, and here's the 2 illumination objectives, the detection objective over here. We would then lift the sample, add the chamber. The chamber has to be filled with about 25 mils of buffer, and then the sample is emerged into the chamber. Okay, so and now could we turn the light down a little bit? Okay. Nope. Nope, okay, that's good. So here's where having a 3D, looking at the whole brain is going to be really critical. If you want to trace neurons from one end of the brain across to the other end of the brain and try and do this with slices of brain tissue, you're just going to have a hard time tracking individual neurons. So this is a whole brain that's has endogenous expression of GFP and TT tomato in different sets of neurons. This is the brain before and after clearing with this organic clearing method called iDisco. And this is a video that I hope will play. There we go. And these are huge files, like terabyte-size files. And so we have workstations set up in the OBC so that you can, we have multiple workstations set up in the OBC. So the data is collected on one microscope and then saved onto another microscope. And so if you're doing tiling, the stitching software is on the second microscope that has a huge, that has a lot of power with it. And then next to that microscope, we have a computer that has Imaris, which we recently got in the OBC. And so Imaris is going to let you do the tracking of the spines and the neurons and everything else, and create these nice movies. So that was the best video I have, okay? Oh, okay, we're going to see it again. No, we're not. Let's go. And so this was taken from Dr. Waterman's lab. George is a student from there. And this sample was cleared with Clarity. We had a demo of an X-Clarity system. Oops. I want to replay this movie. So this sample, because it was a demo and we were in a rush to get the sample cleared and imaged, it didn't get cleared all the way. And so you'll see what happens when it doesn't get cleared all the way. But we're staining. The green is staining for beta-catenin, so it's an antibody stain for beta-catenin, and red is autofluorescence of the vascular system. And I will say the autofluorescence in terms of light sheet actually comes in really handy. So when you're looking at a brain, for instance, a lot of times people will use the GFP, the green channel autofluorescence, to map the whole brain so they can then map it back to the atlas brain. Okay, let's get the movie for this one. So you can see that we didn't get all the way through, but I think we still got a nice movie in here. This is from ovarian follicles. We're looking at oocytes in here, and green are the oocytes. And this takes, the whole ovary takes like 2 to 3 minutes. This is cleared with cubic. And this, you can see, this is actually what we see when we close the door and try and line the sample up and get it all in focus. I'm not going to play this video, because I want to get to the next one. This is a zebrafish. This is a 16-hour time-lapse video, and we're looking at expression of, endogenous expression of GFP and DS red. And they're both expressed. You can see the red is there from the beginning, and the green comes up over time. Again, a 16-hour time-lapse. And you can see why these are like terabyte-sized files. They're massive. So I did really good with time. So if anybody wants to hear about confocals, I can talk about those too. No, I'm just kidding. So if you want to use the light sheet, contact me. The way the OBC core is set up is we're a walk-up facility. I'll train you. The light sheet is completely different than typical microscopy, and so usually, you know, I'm around. I tell people the first few times you come back after being trained, let me know so I can help you get the sample and everything set up. When you put your sample in, the light is going to go through your buffer and then hit to your sample, and you have to make sure everything is lined up right. So you have to play a little bit with the light sheet offset, and so there's different things to play around with. It's not quite as simple as using a confocal, but once you get it going, it's not so bad. There's no charge for training. When you come, you're going to need to bring your sample. Make sure you know what fluorophores are on there. I can't tell you how many people come and they don't know what the fluorophores are that they're imaging. You will need 25 mils of buffer that your sample is in, because again, we want to minimize any changes in refractive index. The recharge rate is $20 an hour. That being said, sometimes image takes, 16-hour time lapse would be a really expensive movie, and so we have a maximum charge of $60 a day. The core is available to you 24/7. Once you get trained, I'll give you a card key, and then you can come in any time. And this is just a comparison of rates from other systems, other universities that have Z1 light sheets.
Thank you.
Okay.
The long time frame experiment, like the zebrafish embryo, how is that orchestrated?
So the zebrafish is mounted in about 0.5% agarose in a capillary, and then the agarose is then just put into the liquid chamber.
So you can move samples in and out over a time period?
No, the chamber, it stays in focus because it's in the low melt agarose, and it's also anesthetized.
So you're just
You just set up the two. You set up the Z stack. Yeah, you just set up the Z-stack and you just walk away. You just tell it from start to finish. Yes.
Have you thought about implementing temperature control so that we can look at mammalian?
So we do have an environmental chamber on there, so it has temperature control and you can have CO2 in there. Yeah. So there's 3 chambers, I should have said that. We have an environmental chamber that has temperature and CO2. You have a chamber for looking at organically cleared tissues, but the objective is never going to come into contact with the organically cleared or the organic material. And then we have one that's a dipping objective for clear samples.
Yeah, during the sample preparation process, will you be able to retain the blood cell, for example, white blood cell that's floating in the blood vessel?
No, the samples are fixed. And
Does that fix blood cells in the vasculature? What's autofluorescing in the vessels?
I think it's the lining. Yeah, probably the collagen.
Collagen?
Yeah,
So it's probably you don't have red blood cells in there or white blood cells?
I wouldn't imagine so.
You do sometimes. Depends if you perfuse before you fix or whether you just fix.
The protocols usually say to perfuse before you fix. So.
That's because erythrocytes will autofluoresce very brightly.
These organically cleared brains, what chemicals do you degrade?
Say that
How do you
again?
turn them clear? How?
You mean, what are you extracting?
Yes.
Is that what-
Lipids.
With
Lipids.
the organic.
Lipids. Yeah, you're getting rid of all the lipids.
Okay, I think we better
Okay.
move along, so thank you very much. So the next talk is about a little known resource, I think, relatively little known. So for people who are interested in transmission EM of tissues, Oz Stewart's laboratory has a transmission EM, which is run as a recharge facility. And he couldn't be here today, but Ilse Sears-Kraxberger, who actually runs the transmission EM, is here today to talk about that. And this is over in Gillespie.
Hi, I'm Ilse Sears-Kraxberger, and I am working at the Reeve-Irvine Research Center in the Department of Anatomy and Neurobiology. We offer biological EM service. We offer consultation for experiment planning, including fixation, sample preparation, and systematic sampling. We prepare the specimen. We will do the embedding, sectioning, and staining, and mounting the ultra-thin sections on EM grids. We then do the EM image collection and also can do a mapping of a location of the images in the tissue. We can assist with interpretation of ultrastructural images, and we offer consultation regarding data analysis. So I'll show you a few samples of projects we've done in the past. So here we did a quantitative analysis of biological ultrastructure, and we looked at synaptic ultrastructure changes in like after nerve injury. So looking at if there were changes in the numbers of the different types of synapses, and also looking at the length of synapses, which you could only do with an electron microscope. Here we assessed consequences of disease and repair. This was a viral model of multiple sclerosis treated with human neural precursor cells promoting neurological recovery. And you can see that there is an increase in remyelination in the image on the right side with the treated animals as opposed to the control. We also looked at G ratio, which is the ratio between the inner and outer diameter of the axons. With a lower G ratio, being an indicator for remyelination, which you can see in the figure on the right. Here we had another case where we looked at axons, demyelination, remyelination, and here on the first image you see like how we did the mapping for the different EM images. So I'm over at Gillespie building, and we can do EM work for you. Thank you. Questions?
So for contact, they would contact.
Oswald Stewart.
Okay.
Yes.
Okay. Thank you very much.
Thank you.
Okay. Thank you all for attending.